Hey everyone, my name is Adam Turner and I am ESR 11 of the BIOREMIA Consortium. This video will be a little bit about myself, a little bit about my project and the PhD that I am a part of. And so my path towards my PhD started with my studying biology at Oxford Brookes University when I graduated in 2016. During this, I gained a broad education in the biological sciences, from environmental science through to biomedical science. And it was here I began to study microbiology, infection and immunology, and discovered my interest and passion for the topic. And as such, I continued to pursue this passion and did a master's degree in microbiology and immunology from the University of Nottingham, from which I graduated in 2017. After this, I spent some time in industry as a biosafety microbiologist before deciding studying for a PhD was where I wanted to go. And so, to begin my PhD project, I moved from the United Kingdom to Gothenburg in August 2020. Which brings me to where I am now, as ESR 11 in the BIOREMIA European Training Network. I have joined the Department of Biomaterials at the University of Gothenburg. And my project is looking at the material-induced inhibition of microbial biofilm formation. But first of all, a little bit of background. Biomaterial-associated infection and infection of bone-anchored implants are becoming an increasingly prevalent issue in the modern healthcare environment. Infections associated with these bone-anchored implants are commonly caused by three notorious clinical pathogens. These are Staphylococcus aureus, Staph epidermidis, and Pseudomonas aeruginosa. And complete eradication of these pathogens, once they've established infection, becomes quite difficult, as soon after they have established, they form these structures known as biofilms. And these biofilms are formed from stable communities of bacteria that are embedded in this slime, this extracellular matrix that substantially enhances their stability. And treating these biofilms is a complex process. It's quite difficult due to both the ability of the biofilm to evade the host immune response, but also the inability of antibiotics to effectively penetrate this slime, this matrix, reach the cells inside and kill them. And so currently the only way to really treat these infections is through revision surgery, long-term antibiotic therapy, or through complete replacement of the implant. Which can be an incredibly debilitating experience for a patient to go through. And even so, treatment can sometimes be unsuccessful and establishment of infection can result in death. As such, there is a need for new materials that can resist the formation of biofilm and prevent infection from ever setting in. And this is one of the focuses of my project. So the main aim of my project here is to assess novel implantable biomaterials, materials that are produced by the other early stage researchers as part of the BIOREMIA European Training Network, and assess their efficacy in preventing biofilm formation in clinically relevant pathogens. These include Staphylococcus aureus, Staph epidermidis, and Pseudomonas aeruginosa, and identify the mechanisms by which this prevention occurs. And so this main aim has been split into three major sub-projects that we intend to complete throughout the progress of this PhD. The first of these is to actually identify biomaterials that exhibit an effective anti-biofilm behaviour that we can then proceed to study further. And then for the second part of the project, we'll be looking to characterise how the materials we've identified during the first stage of our project actually prevents the formation of biofilm. And really dig deeper into the molecular biology and the interactions between our material and the bacteria. We'll do this by studying the structural molecules that make up the biofilm and try to understand the regulation of signal factors that are involved in bacterial communication that's occurring when these bacteria come into contact with our biofilm-resistant biomaterials. For the third stage of my project, we'll be studying the implantable biomaterials produced by our BIOREMIA fellows by looking at how they interact in the context of the host and studying the interactions between inflammatory and regenerative host cells in the same system as the bacteria, culturing them on top or around these novel materials to better understand how they'd interact in a real clinical environment. And hopefully, once we've achieved all three of these stages, we'll have identified the most promising antibacterial and biofilm-resistant materials or surfaces that can be translated into future clinical use. Thank you for watching. I hope you enjoyed. And if you'd like to learn a little bit more about my project and the projects of the other BIOREMIA ESRs, you can find us all up here.